So brain mosaicism is a really relatively new field. And we're discovering that genetically not all neurons or cells in the brain are identical. Genetically, small changes can happen in individual cells during development that make neurons or subpopulations of neurons unique compared to their neighbors. And so it turns out that actually not only in the brain, but all of our bodies, we're mosaics. Not every single cell is genetically identical. And that has evolved from the discovery that certain birth defects of the brain, particularly brain overgrowth syndromes. So there's a whole spectrum of brain overgrowth syndromes that are seen in pediatric populations where either one half of the brain is overgrown, or maybe the whole brain is overgrown, or maybe just a small little feature of the brain is overgrown. And the genetics has led us to the finding that in those regions of tissue or brain that are affected, there are mutations in genes that are really important for pathways that drive normal cell growth and development. And that has led, in the last 2 or 3 years, to a number of groups identifying mutations in the Pi3 kinase pathway, or which is one of the major signaling pathways in the cell. When those pathways are overactive during development, if they're globally overactive, so say you've got a point mutation, or you've got a mutation from one parent, that wouldn't be compatible with life. But if, as a zygote, a few cells in that developing embryo acquired mutations, not all of the cells, but just a few cells, so a mosaic of cells, acquired those mutations, that could lead to overgrowth of that region, or wherever those cells ended up. And it turns out that that pathway is really well described because it's a well-known pathway that causes cancer in adults. Interestingly, apparently when it's active in development, it doesn't cause cancer, it causes tissue overgrowth. So you can have brain overgrowth. If these clones end up in the arm, you have arm overgrowth. We're particularly interested in brain overgrowth. And those brain overgrowth syndromes are megalencephaly, are sort of an extreme case, hemimegalencephaly, or much more commonly focal cortical dysplasia. And focal cortical dysplasia is really important because it accounts for maybe up to 25 to 30% of intractable seizures in pediatric populations. Those seizures are intractable to current anti-epileptic drugs, and the current therapy is surgical resection. So it would be wonderful to be able to avoid surgical resection and have a targeted drug therapy. Now that we know what the genes are that are mutated, that are driving these overgrowths, it's particularly exciting that there is an armamentum of drugs available to treat that pathway, just because that pathway is so well described because of cancer. So in order to test any of those, we need an appropriate place to do that. Obviously, you wouldn't want to be throwing drugs on kids untested, so my lab has generated mouse models of some of these overgrowth malformations, and we have mice with overgrown brains, and those mice have epilepsy just like the humans, and we've started to test drugs that are known for their roles in stopping this pathway in cancer. We've started to test those as a new approach to testing kids, or to treating kids with intractable epilepsy. Sure. And so far it looks like proof of principle. We can overactivate some of these pathways, and proof of principle, first of all, the mice end up with big brains and epilepsy, and we can actually stop the seizure phenotype with some of these drugs. We're very excited that our mouse results suggest that even though these malformations happen during development and the brain structure is abnormal, we're very excited that the drugs seem to ameliorate the seizure phenotype despite the fact that the brain is messed up, basically. And we could only have figured that out in mice. But we're very excited that it means that the therapy is likely a real possibility of therapeutic treatment in humans, and we are very excited that within a few years we will start clinical trials in human patients, and we would be using pediatric populations because that's where this problem is seen in focal cortical dysplasia in particular. In kids with focal cortical dysplasias or even hemimegalencephaly, the best therapy, the standard of practice right now is if they fail at standard anti-epileptic seizure drugs, then the only real possibility for any relief from the seizures is surgical resection of the tissue. And obviously cutting out a chunk of a child's brain is somewhat drastic, and it would be really wonderful to have a targeted gene therapy that's specific for their mutation and active and shutting down the excess electrical activity without having to take a chunk of their brain out. So it is actually tremendously exciting. It's a whole new class of drugs that would never have thought to have any sort of epilepsy therapeutic effect, because who would have thought that this pathway was a major player in epilepsy? So we are making mouse models of these malformations based on human genetic findings in patients with hemimegalencephaly, megalencephaly, and focal cortical dysplasia. So a whole body of human genetic work by neurologists and pediatric geneticists over the last 2 years has revealed the underlying mechanism of these malformations, or the genetic mechanisms of these malformations. Again, these are not your standard Mendelian genetics where mom has one mutation, dad has one mutation, and the kid has 2 copies of bad mutations leading to a disorder. And this isn't even an example of a de novo mutation in the child, de novo mutation in the child, where every cell in the child is affected. It's an even later mutational event where the embryo is formed and then a small subset of cells in that embryo acquired a mutation and continued to grow, so that not the entire eventual person has this mutation throughout their body. There again, it's a mosaic mutation. And it's turning out that these mosaic mutations have been discovered in these brain malformation and overgrowth disorders, but probably mosaic mutations underlie a whole other range of birth defects that we're just beginning to find out about. And it's a really exciting field in genetics, but it's also a really exciting field in therapeutics, because we can model these in mice, develop drug therapies, and then go back to patients. So there really is this cycle of taking human results, going back to the mouse, figuring out the mechanisms of action, drug therapies, and then taking those back to human. It's a whole sort of life cycle. What we're doing is only really possible in mouse models, not even taking cultured neurons in a dish. Epilepsy is a whole disorder of the whole brain, not necessarily one single neuron. It's a disorder of connections and networks of neurons that you can't necessarily recapitulate in a dish. So because we can generate mice with these malformations, they're a much better forum, much better place to be able to test drugs. I don't think we're going to be in therapeutic trials in humans within 5 years. I think there's a lot more work that needs to be done both in humans in identifying the patients and in testing the various drugs in mice. That said, it's very promising that this particular pathway, the Pi3 kinase pathway, is really well studied in cancer. And because it's really well studied in cancer, there are many, many potential small molecule inhibitors that have been developed specifically for therapeutic effect in cancer, some of which are already FDA approved. So leaping to clinical trials in patients for epilepsy, it's not going to be as hard. We're not starting from nothing.